second evening. Why don't you go and have a seat? I'm not going to do the prayer this evening. We have one of our students going to be leading us in that. But I wanted to give you the format. Same as last week. We had such a rave response that we added 2 more presentations. And so next week there'll be even 2 more. So we'll have 11 next Wednesday. We'll just keep adding on to those. Same format. We're going to do a few presentations, take a break, and then come on back and finish up. So Ainsley Joyce is going to lead us in prayer, and she will be our first presenter talking about oysters after the prayer.
Okay, in the name of the Father, and the Son, and the Holy Spirit, amen. O Lord, giver of life and source of our freedom, we are reminded that yours is the earth and its fullness, the world and those who dwell in it. We know that it is from your hand that we have received all that we have and are and will be. Gracious and loving God, we understand too that you call us to be stewards of your abundance, the caretakers of all you have entrusted to us. Help us always to use your gifts wisely and teach us how to share them generously. May our faithful stewardship bear witness to the love of Christ in our lives. We pray with grateful and full hearts. In Jesus' name, amen. St. Robert Bellarmine. Pray for us. Sorry, bear with me here.
Remember that number, 7561. Check your tickets later.
Okay. Just make sure it's up there. Okay. The Gold Rush. The tourists were drawing nearly 300,000 to the Pacific Northwest and the Western United States. The Gold Rush brought 300,000 people to this area, but it's also responsible for the 98% drop in native Olympia oyster populations. The Gold Rush was a period in the mid-1800s that led thousands of people to migrate in search of wealth. It caused rapid population and economic growth, but it also brought environmental changes. San Francisco, namely, represented the largest market for Olympia oysters. Between the years 1851 to 1915, European settlers removed an estimated 5 billion oysters from Walapa Bay, an estuary in Washington state. While overharvesting was brief, the damage was already done. Today, researchers are looking for ways to restore the Olympia oyster. A 2008 study in Chesapeake Bay showed that chemical signals released by adult oysters can induce larval settlement. In other words, the presence of adults actually encourages the recruitment of larvae. If this effect occurs in oysters native to the eastern United States, it may also apply to oysters native to the Pacific Northwest. This brings me to my project. Hi, my name is Ainsley Joyce, and I study the effect of the abundance and proximity of adult Olympia oysters on larval settlement patterns. So, although the gold rush and overharvesting caused the original decline, non-native species such as the Pacific oyster are now what limit Olympia oyster recovery. The introduction of eastern oysters to the West Coast via railroad transport also brought up the predator. The predator's name is the Japanese oyster drill. Depicted on the far right, this oyster drill quite literally drills holes into the bivalve flesh of Olympia oysters. So since the 1970s, Washington State has actually recognized Olympia oysters as a threatened species. And in 1998, the Washington Department of Fish and Wildlife drafted a recovery plan, but unfortunately this was never funded, so that fell through, leaving current researchers to revert back to methods such as replacing shells into the oyster bed and bringing hatchery-raised oysters from aquaculture into those beds. This leads me to my problem statement. There is a gap of knowledge regarding the correlation between oyster larvae and settlement and proximity to the oyster beds. My objective was to determine if there was a relationship between Olympia oyster settlement patterns and their adult presence. My hypothesis was that oyster shells 20 meters outside of the oyster bed would have smaller recruitment numbers than opposed to groups directly in the oyster bed, close to the adults. Throughout this experiment, my role as student was to assemble the shell strings, swap devices each month, and examine juvenile totals back at the lab at Pacific Lutheran University. My mentor, Dr. Behrens, guided the statistical analysis and accompanied the device assembly. He also accompanied me through every single stage in this process. Okay, and on the left is a map of the testing site. So this area is just past the Purdy Bridge in Gig Harbor. It's a beautiful site. If you look on a summer day during low tide, the beach is covered in oyster shells. I've really never seen that many oyster shells in my life, but it's managed by a commercial company, which is why there's so many. They harvest the oysters there. And it was just a really pretty sight, and I'm so grateful I got to venture out there about once a month during the summer. So in order to measure the Olympia oyster settlements, a shell string was created, and this was used to have the oysters settle on a surface because they're made of calcium that actually attracts the oysters. The shells were curated from a different study, and it was comprised of 10 shells, with the 11th shell on the bottom being the barrier between the mud and the shell strings. Okay, and here are the conditions of each of the 4 groups. So groups 1 and 2 are located in the bed, but on opposite ends, so those are the ones close to the adults. Group 3, the control was isolated just outside the Olympia oyster bed. And then group 4 had 200 oysters artificially moved near the area. The group was outside of the bed, but had a couple oysters near it. And so here is a map of the formation. On the left is a map, and then on the right is an image of group 4, which the Olympia oysters are in the center. So a common question is, well, when did these swaps occur? I already said that they do. We went and swapped them once a month. But it was really dependent, the timing, on the low tide. So it needed to happen between -2 and -3 low tide, which is why we went anywhere from the early morning to late afternoon. So following my data collection, my mentor and I brought the shells back to the lab and looked through every single shell. There was about 120 shells, and we looked through that every single month. Typically, I circled what I thought was larvae, and then he either confirmed or denied if that was what I was actually seeing. And Olympia oysters were not the only thing we found on the shells. We found Pacific oysters, which look really similar but are a bit more of an oval shape. We found egg clusters from different species, as well as mussel larvae. Okay, so moving on to the results. So for my results, I conducted a 2-way ANOVA test with the treatment as the effect. The 2-way ANOVA test is a statistical test used to find the difference of 2 independent variables and to see how they affect a single dependent variable. And after the ANOVA analysis was conducted, a p-value of 0.91 was the result. And understanding that a p-value of 0.01 to 0.05 is optimal, you can see that the results are completely out of that range. So contrary to my prediction, the data across groups 1 and 4 vary throughout all months, with no significant outcome across each bed, which means there's kind of no easy way of telling whether or not the adults actually do induce the larval settlement. The first two values on the graph represent the groups that were inside of the oyster bed, so directly adjacent to the adults. And so I hypothesized that those would have the greatest number of larvae. The third value is the control, which was completely isolated, and that actually had the highest. And then the last, the oyster one, which had the 200 oysters, was about in the middle. And this was averaged off of each month. On the other hand, there was a clear correlation with seasonality. This shows that Olympia oysters are in good health and are continuing to reproduce on schedule. This has a p-value of 0.08, so just outside of that optimal range. In June, those had the lowest numbers because it was the beginning of the reproductive season. And then in August, those had the highest numbers because that was peak reproductive season. Because there's a difference in that, we know that it's reliable data, and which is why it has a lower p-value. Okay, moving on to my discussion. So my hypothesis predicted that larvae counted on shells, that it would be dependent on the adults based off of a 2008 study that looked over Sumina oysters. However, I did not find this. And then also it's important to consider that this was a pilot study. My experiment, this was the first year doing it, so it also was adapted to the Pacific Northwest, not on the East Coast. So the big question is, what do these results mean, and how can it be applied to the restoration effort? Well, knowing that oyster settlement does not rely on the presence of other adults, researchers can continue to work through methods that do support recruitment by placing shells in oyster beds and transplanting hatchery-raised oysters. Of course, there are some limitations. So data was collected from a single oyster bed, meaning that results could differ across Washington State or the West Coast and various other oyster beds. In addition, the settlement surface varied along each shell string because Pacific oyster shells differ in shape and size, and so random selection did happen, but as you can see, some are rounder with a larger surface area and some are more oval shaped with a smaller surface area. Standardizing these shell dimensions would improve experimental control, but it's not necessarily environmentally accurate. So as a pilot study, future research would extend to sampling into following summers, increasing the number of shell strings experimented with, and then also expanding into different sites. The blue dots on the map represent potential or already known oyster beds, so that indicates that there is a lot of potential for the future. A big thank you to my parents, Dr. Behrens, Mr. DeGroot, and the Marine Chem class of 2026. You guys have been amazing, and I'm really going to miss this group. Are there any questions?
Way in the back.
Okay, sorry, I can't see very well. Way in the back.
So you think at one point you were talking about like larval recruitment,
Sorry, could you repeat the question?
like at some point you were talking about I think like larval or oyster recruitment. What is their recruitment location?
Okay, so I think the question was, at some point I was talking about larval recruitment. What does larval recruitment mean? So great question. That essentially means, I'm going to go back to the slide so I can show it. Okay, so these shell strings, these are placed into the water, and as the tide flows, larvae are supposed to settle onto these shells. So larval recruitment means that the early stage Olympia oysters are going to be settling on this, and the purpose of the study is to find out how we can increase the number of Olympia oysters that settle in a given area. And so we're trying to increase the recruitment, the larval recruitment, by having the adults near. Great question. So the question was, I mentioned in my limitations the different sizes of oysters. Could I be able to drill down to like make them the same size? My mentor and I actually talked about crushing the shells and putting them into a mold to have them all be like a square tile. But ultimately, we kind of came to the conclusion that it could mess with the chemical makeup of it. Of course, it's made of calcium, but that could be a potential option for future research. We just never got to it.
Come back next year, you'll see those results.
Yes, come back next year. Yes, in the back.
Temperature or bacteria affect the growth?
So the question is, does temperature or bacteria affect the recruitment? So actually it does. Going back to my results slide, August has the highest number of recruitment because the water is much warmer then. Even in June, even though it's technically summer, the water temperature is lower and it's not quite ready for the oysters to settle. So temperature does play a big role, and it increases the settlement of the oysters. Okay, thank you. Next up, we have Riley Conklin and Luke Ziegler presenting on fruit flies.
All right, you might have heard the statistic before that cancer is the second leading cause of death in the United States. That's after heart disease. But what if we could lower those statistics and make cancer something that's less prevalent in our society with something as simple as like a dietary change, something that you might like to eat anyway, like coconut or pomegranate. Hi, I'm Luke Ziegler.
And I'm Riley Conklin.
Today we'll be presenting to you on our project, observing the expression of, or assessing the effects of dietary antioxidants on the developmental timeline of Drosophila. Mr. DeGroat told me this before we presented. We've been saying it wrong for the last 8 months. Drosophila melanogaster with the RAS oncogene. So first up, I'm going to give you a little bit of background on what ROS antioxidants are. So first up, we have reactive oxygen species, or ROS. These are small hydrogen-based molecules, called free radicals also, which are missing an electron in their outer shells. Because of this, they steal electrons from things around them, because they want to become balanced again. And this damages cell structures near them, as well as DNA, according to Mishra et al. They're naturally produced by organelles, especially the mitochondria, to kill damaged cells, but if they're produced in large numbers, they can be produced in large numbers by outside factors like radiation and high UV exposure. So what happens if you have a large amount of ROS in your body? Oxidative stress. It's a condition that cells experience when there's an overabundance of ROS. This is something that could happen if you're exposed to those outside factors. Oxidative stress damages healthy cells and DNA, and in a study done by Schwartz et al., it was found that ROS concentrates around the mitochondria and especially affects your cellular energy levels. But how does this relate to cancer? When cells remain in oxidative stress for too long due to excessive ROS, it can damage cellular components such as DNA, proteins, lipids. But especially the DNA matters because that leads to potential cell death even in healthy cells. But even worse, if DNA is damaged by the ROS but the cell survives, it can sometimes be modified into a cancerous state, according to our sources. This is important, especially to mention that ROS is not the cause of all cancers, but it is partially responsible for many. So now you know what oxidative stress is, you know what ROS is, and how that causes cancer, but how does your body counteract that? Because we're all exposed to UV every day, right? One way to neutralize ROS is by ingesting foods rich in antioxidants. In theory, dietary antioxidants work because they are molecules which are stable enough to donate an electron to a free radical and neutralize it. You can see in the diagram on the screen, an antioxidant has lots of extra electrons and can afford to donate several to different free radicals to neutralize them. They're also found in a lot of foods like spinach, pomegranate, coconut, and sweet potatoes.
Let me introduce you to our test subjects. We conducted our study using Drosophila melanogaster, but we'll just be calling them fruit flies for the rest of this presentation. Mr. DeGrow would not let us test on humans because the guidelines for their captivity were too complex. Nearly 85% of human disease-causing genes have homologs in fruit flies, which allows for accessible cancer research according to Yamaguchi. Additionally, fruit flies are affected by free radicals and antioxidant balance in their genetics and diet. With fast reproduction cycles and large outputs of offspring, along with their short lifespans, they can be an ideal test subject for many studies. Our flies were genetically modified to have high ROS levels, which we accomplished with the RAS oncogene. The RAS gene is a gene that everybody normally has in their bodies that controls your body's production of ROS. The RAS oncogene is a mutated version of the RAS gene, which our flies have been genetically modified to have. This version that our flies have is permanently activated, causing their ROS levels to skyrocket. It's very similar to a light switch. Our problem statements are that it is unknown if the effects of high ROS can be mitigated by the introduction of dietary antioxidants, and if this could lead to a larger population in fruit flies with elevated ROS. High ROS can decrease the overall health of an individual and lower their cellular energy levels. Our objective is to examine how dietary antioxidants affect the percentage of fruit flies that survive to adulthood. Our hypothesis was that flies given the antioxidant beverages will see a higher percentage of their offspring survive into adulthood.
So how do we accomplish our study? First up, as Riley explained, our fruit flies had something called the RAS oncogene. This is the light switch gene. They gave our flies a constant level of high ROS. You can see here the specific gene written out as you see in their genotype. And remember, this is important because the sort of ROS, or the sort of conditions that would cause us to have high ROS are not easily replicable in a lab. Like, we couldn't irradiate our flies to give them high ROS. So instead, we did it genetically. So we designed our experiment around 3 trial groups. The trial groups had 2 vials each and in pairs. They were treated with either water, 1-to-1 coconut water and water, or 1-to-1 pomegranate juice and water. We used a system where we put 3 males and 5 females into test tubes with their assigned variable from the start of testing to after the 20-day mark when they were removed. The vials were then left alone over a period of 45 days, and all the young that emerged from that time were observed. To keep our flies alive when we weren't testing on them, we fed them words, Drosophila medium mixed with water, and they were kept in a sterile environment with tubes containing that medium and the given antioxidant beverage. For breeding, again, 5 non-virgin female flies were selected and paired with 3 males, which appeared to be in good health every time we needed a new vial. And overall, we just did good general upkeep, and we threw out any vials that had mold or rise, which I'll talk more about later. So for our analysis, to collect data, we counted the offspring once they reached the end of the 3rd instar phase. There were 3 critical phases that we focused on. The 3rd instar phase is when the fruit fly larvae crawl up the side of their vial and prepare to pupate. And when they do that, when they pupate, we counted them again and cataloged them in our data sheets. The final time they were cataloged is when they emerged as adults. And the studies we looked at showed that the most critical portion of their life, especially for their mitochondrial energy levels, is in that pupation to emergence phase. So here's what we found. Let's take a look at the data for our objective survivorship. So here you can see on the graph, I have laid out the total number of larvae versus adults according to the incubation period, or accounting for incubation period. If you guys have questions about that, ask me after. So here in the gray, you can see the number of pupated larvae that we had at the end of the 45-day period. That's the mature ones, the ones that had enough time to become adults if they were able to. And then in the yellow, you can see the total adults. You guys can't see that, right? Yeah. And in the yellow, you can see the total adults that we ended up with at the end of the 45-day period. Surprisingly, we had a large amount of larvae in the control, which we weren't really expecting, but unsurprisingly, in the coconut and the pomegranate, you can see that even though the larvae was lower, we actually had a much more successful level of adults successfully become adults. So, again, the fly population for just the larvae was kind of weird. Here in the notable characteristics of the data section, you can see that our data displays trends when averaged, like you can see on the last slide, but it was widely varied in population size overall. I mean, in the control alone, which is in blue here, you can see that there was a difference of over 100 individuals, despite the ratio of those that survived to those that remained pupated being about the same.
Moving on to our discussion. As you might recall, our hypothesis was that the flies, given the antioxidant beverages, would see a higher percentage of offspring surviving to adulthood. Once we compiled all of our data and compared the vials and tests to one another, our data suggested that our hypothesis might be correct, but additional testing is required. Our ANOVA, or Analysis of Variance test, resulted in a final value of 0.1794, which indicated that our data does not carry statistically significant weight. However, many of our individual data points collected did show correlation between the diet and overall health, and we suspect that our statistical range was off due to the small sample size. Additionally, when two population data points are very far apart, it is known to skew the ANOVA results. As you can see with our percent survivorship, which was used in the ANOVA test calculations, the percent for two trials can differ greatly. Namely, the coconut vials, where one vial saw a 49% survivorship rate and the other saw an 89% survivorship rate. ANOVA tests are typically used for a range of data to test a hypothesis among groups. However, we only had 2 values for each diet because of the initial problems and time restraints with our project. We suspect that if the test was done with more trials, they would produce an actual range of data and the project would be statistically significant. So even though our data for the survivorship and offspring was deemed not statistically significant, if you look at the raw data, you can see a large difference between the amount of offspring to survive to adulthood among our test groups. The blue arrow points to the control group's survival rates, and the orange points to the survival rates of the pomegranate group. Clearly, the groups given antioxidants were much more likely to survive through pupation. This signifies that it's likely their mitochondria worked well enough to keep them healthy to survive the pupation process. Because the mitochondria produces ROS and energy, if the ROS levels are too high, a fly will not survive pupation. More flies surviving pupation when given antioxidant-rich diets signifies that the diets may have an impact on balancing ROS levels and potentially mitigating cancer in the flies. So diet could truly potentially help with imbalanced ROS and reduce cancer rates for people with higher ROS levels from UV radiation, certain genetic predispositions, and DNA damage. Due to the nature of our project, we had many limitations on our research. The condensed time frames and limited fly populations meant we were unable to use adults that were all the exact same age in our project. We could not tell when a fly was anything other than very old or very young, so we kind of had to guess that they were of an age to reproduce. Older flies might have produced more offspring right off the bat and died off faster, while younger flies are likely to produce more long-term but take longer to initially start producing. Additionally, most likely due to the potential difference of other adults, of the adults in age, some vials produce more larvae than others, which may have led to some statistical error. Because we could only do 2 tests per diet due to time constraints, doing the statistic test was very hard, and we did not have enough data points to provide a full range for each. There were also some smaller issues we faced in our project, like reef mold issues, the flies deciding to pupate the food source, which made counting them really difficult, and making sure the flies had the right genes, because there was some potential early on for some cross-contamination.
All right, so raise your hand if this project was absolutely enthralling and you'd like to do some future research. Oh, I see a lot of hands. You guys can't do any more research. You're done. Yeah. All right. So if for some reason you actually thought this was really cool, then what could you do? First off, there are a number of places to go from here. Future research could be focused on the specific effects of the antioxidant diet on organs like the gut, which we study because it's the only organ in a fruit fly that's big enough to study. And you could also look at the lifespan of adults after they emerge. We didn't do that because we just killed them. This could help to establish a complete survivorship curve. Also, obviously, we didn't test spinach or sweet potatoes, and if you're a fan of those things, you could test that.
Thank you to Dr. Leslie Saucedo for teaching us how to take care of the flies, how to understand genotypes, and for just generally helping us to form our project. Thank you to Mr. DeGrow, our families, and to Caroline and Tessa for supporting us in various ways throughout this project. And thank you to the class of Marine Chem 2026 for all the memories.
You guys are awesome. Thank you.
Are there any questions? Her question was, how many flies died and what happened with the mold? Because we were taking care of the fruit flies for roughly 8 months, and they die every 3 weeks, and we had thousands at a time, I would guess, like, maybe 6 digits of flies died, or maybe 5 to 6 digits of flies died during our project. But many of them lived their full, unhealthy lives. Yeah. As for the mold, if you leave fruit juice out on the counter for 45 days, it doesn't want to last the whole time. So it got moldy, and the mold would instantly kill the flies, so it's problematic. We figured out, well, we bought a mini refrigerator. Yeah.
Why would the flies have the fruit protein? Why do they have larger numbers?
So the question was, why did the flies that were not fed antioxidant beverages end up with a higher larval number? Good question. Originally, that was going to be another one of the things that we were looking at mainly, but we really don't have an answer for why that was the case. We sort of have been wondering about this, and one of our possible ideas for why that was the case is that either it was luck with the ages of the flies we put there to start with, or it might have had something to do with the levels of sugar in the beverages. We don't know. That's just total guesswork, but good question.
The question was why we chose fruit juice instead of actual fruit. And the reason why is because the fruit flies we had were bred in captivity, and they only know how to eat this very specific blue mush that is prepared with water and this dust that turns into the blue mush. And it's pretty easy to replace the water with fruit juice, but we didn't know how to incorporate the actual fruit into the blue mush.
You there? Because I like coconut water and pomegranate juice better. Oh, sorry. The question was, why did you choose coconut and pomegranate instead of something else that was more boring? And the answer is because I like coconut water and pomegranate juice better.
And another answer is that in health media, coconut and pomegranate juice commonly promote themselves as very healthy antioxidant drinks, and we wanted to test if that was actually true.
That was a better answer.
The question was, how did we acquire the flies? The answer is, we basically got them from Caroline and Tessa, who did a similar but different project with them, who got them from our mentor, who we shared with them. And the mentor was originally using them for a very similar project, testing antioxidants in cancer, but in different ways. And she said that most scientists actually don't really know the correlation between antioxidants and balancing ROS levels, and that's how we decided to use them for a project.
Do we You guys done? Thank you guys. All right, next up is the wonderful Emily Robinson presenting on how oxygen affects you when you swim underwater. I think. Let's give her a hand.
2 minutes. That is the average time it takes a person to black out in response to a lack of oxygen. 1 to 3 minutes is the typical range. 120 seconds can be the difference between life and death. Hi, my name is Emily Robinson, and my project is on assessing the risk of hypoxic blackout in synchronized swimmers. When most people think of synchronized swimming, they picture girls in flower swim caps floating on their back. While that once was the case, the sport has evolved greatly over time. It is now a highly competitive sport that is physically demanding. Athletes push themselves to the limits of breath holding to gain a few more points. As of a few years ago, the sport has become more centered around being underwater, as the more you do under, the more points you receive. This has led swimmers to push themselves beyond their limits, to the point where they pass out underwater. This poses a dangerous threat to the athletes and is not just common in synchronized swimming. Freedivers often do this as well, as the whole point of their sport is to hold their breath. And although these sports are fun, safety concerns need to be addressed, as there are many dangers that come with them. The idea for this project stemmed from my own personal experience with this as a synchronized swimmer myself. A few years ago, at practice, a teammate of mine suffered from a shallow water blackout while swimming laps underwater in a common training set that we would do, almost costing her life. This incident opened up my eyes to the dangers that this sport poses and inspired me to become more educated on them. So what is a hypoxic blackout? Well, hypoxia is a condition in which there is an inadequate supply of oxygen to the brain, to the body's tissues. One of the main types of hypoxia is hypoxemic hypoxia, which occurs when there is not enough oxygen in the blood. Symptoms of hypoxia can vary depending on the situation and severity, but can include headache, dizziness, fatigue, rapid breathing, and a syncope, or fainting underwater. When swimming, shallow water blackouts, or hypoxic blackouts, are common. This is a loss of consciousness due to a lack of oxygen to the brain during a breath hold. This is often made worse through hyperventilation, which lowers carbon dioxide levels, decreasing the body's natural urge to breathe, leading to a syncope, or fainting. These do not just have to occur in shallow water despite the name, but these are a common way that drownings do occur. And this can happen to anyone, whether they're an athlete or not, and is a silent risk. It is not like an active drowning where the person is moving. They may seem fine right before they black out, making it even more dangerous and harder to spot. Synchronized swimming involves high levels of aerobic and anaerobic activity, meaning that the sport involves actions that use up a lot of oxygen and some that don't. Aerobic stores are normally considered the major determiner of breath-holding ability. When the body is not getting new oxygen, it relies on the stores already present, and improving aerobic capacity can increase breath hold duration. Lactate is used after exercise and recovery for resynthesizing. When athletes hold their breath and exercise, lactate will accumulate faster. In synchronized swimming, the short breaths allowed throughout the routine result in lower lactate levels than in disciplines such as freediving, where there is no breath allowed. Overall, blood lactate is influenced by the duration and the type of work, and in synchronized swimming, the duration is shorter, so less lactate accumulates. Previous studies have shown that holding your breath underwater initiates a diving response. This means that the heart rate slows and blood pressure increases to help the body conserve oxygen. This slows down the loss of oxygen to protect the body, as it is not getting new oxygen but still using the original supply. Oxygen saturation, also known as SpO2, which is what I will refer to it as, is the percentage of oxygen-carrying hemoglobin in the blood compared to deoxygenate. For most adults, the ideal range of XPO2 is between 94 and 100%, but 85 to 92% is common in adults with chronic lung disease. If the SpO2 drops to 85 to 93% in a healthy adult, there is a high risk of hypoxia, and that risk only increases as the oxygen drops below 85%. When the SpO2 levels are this low and the heart rate drops as well, there's a very high risk for hypoxia and a loss of consciousness. And if the heart rate is unstable, in tachycardia or bradycardia, a hypoxia-induced syncope, arrest, or seizure is very likely. This leads me to my problem statements. There's a gap in knowledge about how synchronized swimming training methods create the risk for hypoxic blackout, and there's also a gap in knowledge about how exercise while breath holding compared to static breath holds affect oxygen saturation. My objectives are to assess the risk of hypoxic blackout in synchronized swimming training exercises and to compare the effects of exercise and static holds on SpO2. My hypotheses are that these training methods will result in a high risk for hypoxic blackout due to the drop in SpO2, and that with increased exertion from exercise, blood oxygen will decrease faster, therefore increasing the risk for hypoxia. My experiment aimed to monitor SpO2 and heart rate in typical synchronized swimming training methods. An Oxline Pulse MD Pro was used to measure the SpO2 and heart rate. In this device, a subject would simply stick their finger and a measurement would be given of their heart rate and SpO2. In order to get an accurate SpO2 reading, the device would shine a red and infrared light into the finger to measure the amount of oxygen in the blood. It can calculate this by finding the ratio of oxygenated hemoglobin to total hemoglobin based off how much light is absorbed. The athletes participating were chosen from a local synchronized swimming team and include myself. There were 7 athletes total, all females ages 13 to 18 who have similar training schedules. 3 tests were performed, and tests 1 and 2 were very similar. In test 1, swimmers performed 4 25-yard laps of no-breath freestyle with minimal rest in between each lap, meaning they would swim one length of the pool without breathing, and then I would record their SpO2 and heart rate, and they would repeat this a total of 4 times. Test 2 was the same, but instead it began with a 100-yard individual medley swim before the 4 laps of no-breath swimming. During this 100-yard swim, the athletes were allowed to breathe as necessary. The goal of the second test was to see if starting the no-breath laps with a higher heart rate would have an influence on the SpO2 levels. The third test was a static breath hold, where swimmers held onto the wall while they submerged themselves underwater, wearing the device on their finger continuously. They remained underwater for 60 to 90 seconds or until they were too uncomfortable to continue. Their heart rate and SpO2 levels were measured every 15 seconds, and the purpose of this test was to compare oxygen levels with exercise to levels without exercise. These 2 graphs show the results of the first test, the 4 laps without breath. Both of these graphs show averages of all of the athletes to create a general trend, and this will be how all the graphs that I show you will be. Each dot represents a data collection after each lap. The graph on the left shows how the heart rate increases a lot during the 4 laps, while the graph on the right shows how the SpO2 declines. Overall, the SpO2 declined from 95.3% to a low of 85.8% in this test, while the heart rate increased to a high of 166 beats per minute. And a similar thing happened in the next test, the individual medley, followed by 4 laps without breath. But this time, a rapid drop in oxygen occurred before an incline at the end. In this test, the first dot represents the data after the 100 IM, and the other 4 dots represent the data after each no-breath lap. The average heart rate followed a similar pattern, but the 4 laps no breath started at a higher heart rate than expected, or as expected, as the 100 IM was performed before the 4 laps. The graph on the right shows the oxygen levels. The SpO2 again declined consistently at the beginning, but then had a sudden drop after the 3rd no-breath lap. This is due to the different levels for different people. A wide range of SpO2 levels were recorded, and these graphs again show an average. In the last test, the heart rate dropped drastically, which is shown on the graph on the left, due to the static nature of the test. This was as expected. You may notice that the first heart rate recorded, which was basically the athlete's resting heart rate, was abnormally high. This is due to test anxiety before completing the set. It reached a low of 49.3 beats per minute, significantly lower than the other tests, and an overall heart rate lower than normal. SpO2 also declined to a low in this test. The lowest level reached was 85.5% in the high risk for hypoxia zone. Overall, my hypotheses were partially supported. Test 1 and 2 had similar results, with the heart rate increasing as the SpO2 decreased, but Test 2 resulted in higher SpO2 levels than Test 1, contrary to what I expected. This shows us how starting with exercise and a higher heart rate prior to breath holding does not increase the risk of hypoxia by lowering the SpO2 levels. In Test 3, both the SpO2 and heart rate decreased significantly. As I mentioned, oxygen reached a low of 85.5% in this test. This is due to the duration of breath holding compared with the other tests. Heart rate also reached a low of 49.3 beats per minute in this test due to the static nature. This shows us how the static breath hold initiated the diving response that I mentioned earlier. The athlete's heart rate slowed drastically to conserve the oxygen already in the body as the body was not receiving any new oxygen. This shows us how the duration of breath hold has a greater effect on oxygen levels than exercise itself. Exercising while holding one's breath does lower oxygen levels, but not as much as a consistent breath hold. So what can we conclude that this means for the body? The lack of oxygen in synchronized swimming exercises causes the body to produce energy without using oxygen. This also increases the amount of lactate, which leads to increased fatigue. When there is low oxygen in the body, more lactate is produced than can be broken down by the body. And all of these factors, combined with the low oxygen levels from our results, show how synchronized swimming training methods affect the body negatively in its systems during exercise. There are many limitations in this study, including that the athletes used were all from one club and had similar training schedules and skills. They were also all females of similar ages, as I mentioned, and only a few subjects were used. Another limitation was that this project was not my original plan. I had first reached out to people who were doing similar research in Sweden on freedivers, and although I was invited to help them assist in their lab, unfortunately I was not able to go, so we decided on this, a more local and personalized project. Future research could be conducted to see what causes the different levels of SpO2 in each person, as well as new training techniques can be created to create a safer environment. Whether this means consistently monitoring factors such as SpO2 or putting breath hold limits on routines, there are ways to implement precautions into the sport. So what does this tell us about synchronized swimming as a whole? Training methods lead to unhealthy oxygen levels, therefore changes need to be brought about. Athletes are training themselves to resist the urge to breathe, putting themselves in dangerous situations. Synchronized swimming, along with other breath hold sports, need to implement safety precautions and put limits on breath holding so that no athlete is put at such a high risk for hypoxia. This needs to be shared and realized by athletes and coaches alike so that they can adapt training to create a safer environment. I want to give a big thank you to Mr. DeGroat for all of his help and guidance in this project, and of course my teammates who were generous enough to be a part of this experiment, and my friends and family for their constant support, and of course the Birmingham class of 2026 for all the fun memories. My references. Are there any questions? Was the question, what was the average time for the lap swims? Yes, okay. I would say it was around 20 to 25 seconds. Yes, in the back.
Did the athletes swim multiple rounds of the 100 IM followed by the 4 no breath laps, or just once?
So the question was, did the athletes swim multiple rounds of the 100 IM followed by the 4 laps no breath, or just once? And it was just once. So the athlete would simply swim the 100-yard IM, and then I would record their heart rate and SpO2, and then they would immediately go to the 4 laps no breath.
So I have a follow-up question. Do you think that having multiple rounds would compound the data that you saw with the SpO2 levels?
So the question was, if having multiple rounds would compound with the data that I found, compound with the data and change the results. That's a great question, and I feel like that's a good route for future research. I'm not sure. There could be correlation with that if more exercise would lead to a drop in oxygen saturation, but that's definitely an option for research. So the question was just a clarification on what the laps meant, like 4 laps, no breath. So it was, they would swim one length of the pool, and then I would record their heart rate and SpO2, and that would give them about like 5 seconds of rest. Then they would immediately go into the another lap of the no breath freestyle, and they would just repeat that 4 times.
And then another question was that testing these, is that similar to sets that the synchronized we have during the practices? And probably during the
So the question was, is this set like a typical training routine or exercise for synchronized swimmers? And the answer is yes, this is something that we do quite often, but I just wanted to see if there was a risk for hypoxia in that. Yes.
You mentioned that one of your limitations was that you're like don't study with all the other girls. Obviously you didn't research this, but in your opinion, do you think having boys also do this would change your findings?
Question was if having boys in my study would have changed my findings. I don't think that having boys would change the findings, but all the subjects were of similar age and skill level, and they were all girls, but that's definitely an option for further research. But there's not, unfortunately, there's not a lot of boys synchronized swimmers to test on, so that was an limitation. And next up we have Anna Shimshock, who's going to talk to us about cloning.
Hi, my name is Anna Shimshock, and today I'll be presenting my research titled Predicting Somatic Cell Nuclear Transfer Embryo Success Using Gene Expression. This project was completed with mentorship from Stephen Shimshock. In this presentation, I'll explain why cloning procedures fail due to early gene expression abnormalities, how I used machine learning to quantify embryo normality and predict developmental risk, and how this can improve cloning efficiency while reducing economic loss and animal welfare concerns. Somatic cell nuclear transfer, or SCNT, is a cloning technique where the genetic information of a body cell is transferred into an egg cell. This process works in 4 important steps, as shown in this visual. First, the nucleus from the donor cell, containing all its genetic information, is removed. Secondly, the nucleus of the egg cell is removed to prepare for a new nucleus. Third, the donor cell's nucleus is inserted into the egg cell, often via electrical pulse. Lastly, the embryo then uses the new DNA to start development. This process famously produced Dolly the sheep, proving that adult cells can, under the right conditions, be reset to support full development. Scientists utilize the process to advance medical research, improve agricultural efficiency, conserve endangered species, and study genetic processes. However, while SCNT works occasionally, especially in controlled lab environments, it is an extremely inefficient procedure with a success rate of only 10%. Most failures occur very early in development, often before or during implantation. These failures lead to significant economic losses, high pregnancy failure rates, and serious animal welfare concerns. Importantly, these failures are not random. Research shows that they are caused by molecular reprogramming errors, where the transferred nucleus does not fully reset its gene expression patterns. A foundational study done by Bias et al. In 2016 examined gene expression in cattle embryos produced by SCNT and artificial insemination, or AI, with AI embryos serving as the normal developmental reference. The researchers analyzed 2 key stages of pregnancy, day 18 and day 34, to capture the transition in implantation, a critical checkpoint where the embryo must successfully attach to the uterus and begin placental development. At day 18, just before implantation, SCNT embryos showed widespread transcriptional disruption, with over 5,000 genes abnormally expressed compared to AI embryos. By day 34 after implantation, the surviving SCNT embryos appeared more genetically normalized. However, this apparent recovery coincided with abnormal gene expression and stress signals in the maternal uterus. These findings suggest that early molecular defects in SCNT embryos are not resolved, but instead manifest later as disrupted embryo-maternal communication, ultimately contributing to developmental failure. While studies like Bias et al. Are incredibly informative, they are almost entirely descriptive.
You lost your screen.
While studies like Bias et al. Are incredibly informative, they are almost entirely descriptive. They explain what went wrong after failure occurs, but they do not help answer a critical, practical question. Can we predict early on which embryos are likely to fail? Currently, embryologists select embryos mostly based on visual appearance, but genetically abnormal embryos often look completely normal under a microscope. This means that we are missing hidden molecular warning signs. What makes this problem especially challenging is that SCNT failure isn't caused by a single gene or pathway. It's a system-level failure involving thousands of genes that are slightly misregulated. That means traditional approaches that look for one or two bad genes miss the bigger picture. This is why prediction has lagged behind description, and why computational approaches are necessary to capture this complexity. This leads to the central question of my project. Do early gene expression patterns contain enough information to predict how developmentally normal a cloned embryo is? My hypothesis was that early transcriptional data, despite being noisy and complex, encodes measurable information about developmental potential that can be extracted using machine learning. My approach involved three major steps. First, I imported the data from the research done by Bias et al. From there, I used a method called principal component analysis to define what normal gene expression looks like, and quantified how far each cloned embryo deviates from that norm. I then trained a predictive random forest regression machine learning model to identify which genes were markers of success or failure risk. The publicly available gene expression dataset from Bias et al., including both SCNT and artificially inseminated, or AI, cattle embryos, was the foundation of the machine learning model. I imported this dataset into the 9 analytics workspace, which allowed me to build a reproducible, step-by-step machine learning pipeline. 9 handled data pre-processing, dimensionality reduction, and model training without altering the original biological data. To the right is a picture of our data flow in the 9 analytics platform. To quantify how normal or abnormal each embryo appeared, I created a transcriptome fidelity score using principal component analysis, or PCA. PCA takes expression data from thousands of genes and reduces it into a small number of dimensions that capture the main biological patterns. Importantly, the PCA model was built using only AI embryos, which allowed me to define a reference space that represents normal gene expression. Both AI and cloned embryos were then placed into this space. I defined the center of this space as the average gene expression pattern of all AI embryos. The fidelity score measures how far each embryo's gene expression pattern is from this normal center, while accounting for the natural variation seen among healthy embryos. Low scores indicate embryos that closely resemble normal development, while higher scores reflect increasing levels of abnormality. Using a continuous score captures the fact that early development exists on a spectrum rather than as a simple success or failure outcome. In this analysis, expression from roughly 18,000 genes is condensed into 8 principal components, which together captured over 90% of variation among normal embryos. This violin plot shows the distribution of transcriptome fidelity scores across embryo groups. Each violin represents how embryos are spread across score values, with wider regions indicating where more embryos cluster. AI embryos show a tight cluster of low scores, which is expected since they define the normal reference and vary little from one another. In contrast, SCNT embryos display much broader distribution. Some fall near the AI range and appear relatively normal, while many show higher scores, indicating substantial divergence in gene expression. This variability, which is clear on a plot, is exactly what the predictive model is trying to capture, information you can't learn from simply looking at a cell. I then trained a random forest regression model, a type of machine learning model, to predict fidelity scores from early gene expression patterns. Random forests were chosen for several factors. They can capture nonlinear, complex interactions between genes, and the extreme biological noise of SCNT data. Random forests resist overfitting, and they provide feature importance rankings, allowing us to identify the most impactful genes. Model performance was evaluated using threefold cross-validation, meaning the model was repeatedly tested on unseen data. The full transcriptome model explained about 16% of the variance in fidelity scores. While this may sound modest, in a system as chaotic as early embryonic development, this is meaningful. It demonstrates that early transcriptional structure already contains real predictive signal, even though many additional biological factors are involved. To improve interpretability, I focused on the 20 genes most strongly associated with the most predictive principal component. When the model was retrained using only these genes, predictive power improved to about 22% of explained variance. This suggests that a relatively small subset of genes carries a disproportionate amount of information about developmental abnormality. Once early gene expression was shown to be predictive, the next step was understanding what biological processes were driving that signal. Rather than relying on a single gene, the model consistently highlighted groups of genes that work together to reflect how well an embryo is managing early development. Embryos that most closely resembled normally developed embryos showed stronger activities in pathways involved in cell signaling and RNA regulation. This suggests these embryos are better at receiving developmental instructions and handling the intense transcriptional demands of rapid early cell divisions. In other words, they aren't perfect, but they are more capable of maintaining molecular order during a highly unstable stage of development. In contrast, embryos that diverged most strongly from the normal reference showed increased activity in stress response pathways and genes involved in cell adhesion and tissue organization. This pattern indicates early metabolic strain and structural instability, both of which can interfere with proper tissue formation and placental development. Overall, the model is detecting broader biological patterns rather than individual gene effects. These patterns point to three recurring challenges in SCNT embryos: difficulty managing metabolic stress, disrupted control of RNA processing, and weakened tissue organization. Importantly, these signals appear early, before implantation, showing that early gene expression contains warning signs that predict developmental risk long before visible failure occurs. This study has several important limitations. The transcriptome fidelity score reflects gene expression similarity, not direct developmental outcomes like implantation or live birth. The dataset is also relatively small and drawn from a single cattle study, limiting predictive power and general reliability. Despite these limitations, the results show that SCNT failure is not random. Future work should link early gene expression profiles to concrete outcomes such as implantation success or live birth, allowing models to predict true biological success rather than transcriptomic similarity alone. Expanding this framework across larger, multi-study datasets would also improve reliability and generalization. Finally, integrating additional molecular layers, such as DNA methylation or chromatin accessibility, could better capture the reprogramming process. Together, these steps move toward a future of non-invasive, data-driven embryo screening, improving cloning efficiency while reducing cost and animal costs. I'd like to thank my family, especially my dad, Stephen Shimshaw, for mentorship and Bellman Preparatory School for their support, as well as my friends in the Marine Chem class of 2026. Thank you for listening. I'd be happy to take any questions. question was how did we settle on 8 as the number of principal components? So in the KNIME software, it showed that there were 8 groups that kind of all worked together, like those genes all worked together and expressed similar in pattern. So we quantified it into those 8 groups. Yes.
What was the mentor's role in the project?
I believe the question was what was the mentor's role in the project? My mentor, he does data analytics for his job, so he was very familiar with like the KNIME platform and was able to show me how to use like the data flow, and then we kind of went through it together, putting together the data and figuring out the results. Yes.
Who are you going to clone first?
The question is, who am I going to clone first? And the answer is myself. Yes.
Among all of the analytical programs and software, what did you decide?
The question was among all the analytical softwares, why did we choose KNIME? The answer is just that was what my mentor was most familiar with, so it was easiest to kind of use and know how to use. Next up, we have Liam and Enzo talking about Phytophthora.
$5 billion. That's how much money is lost per year to just a single species of Phytophthora, with others adding billions more. This is just the tip of the iceberg with the problem of Phytophthora that we will look at in this project. Hello, my name is Liam Cratley.
My name is Rosa Belcher.
And our project is developing a protocol for the identification of Phytophthora for use by high school students.
Now, why does Phytophthora matters? Now, billions of dollars, as previously mentioned, billions of lives and acres of forest have been destroyed due to the deadly parasite of Phytophthora. Phytophthora has been a problem for centuries, with even being the cause behind the Irish potato famine. Phytophthora owes its persistence to its difficulty to identify, which causes it to be mistaken for other diseases. This prevents scientific communities from properly dealing with the disease in a timely manner, which causes more devastation. Even after the disease is identified, it is still difficult to deal with strong resistance to most disease cures, which prevents blanket treatments like antibiotics from working on the disease. Now, symptoms of Phytophthora are as follows. The contaminations can vary depending on the type of plant. In fruit-bearing plants, symptoms can manifest themselves through wilting, softening of fruit, and tissue collapse. Phytophthora has a more devastating effect in human conditions, as plants often become more susceptible to being infected by Phytophthora. In woody plants, on the other hand, symptoms are more gradual and often harder to detect. Some common symptoms can include root rot, cankers, crown decline, but signs of infection may remain latent for months or years. Oftentimes, Phytophthora is detected too late in plants, as they can be easily confused with other plant diseases. Now, what is Phytophthora actually? Now, Phytophthora is a protist that is classified as a parasite, which is a water mold that thrives in water and soil environments. It can remain in the soil for a long time before infecting a host. Thus, Phytophthora is often only identified too late, allowing it to cause severe damage in crops, preventing a major issue for the agricultural industry and natural ecosystems. Phytophthora also spreads through water, runoff, soil, contaminated equipment, and even animals, easily infiltrating new environments. In addition, climate change has created more stressful conditions for plants, so they cannot be as defensive against parasitic pathogens such as Phytophthora due to weakened immune systems. This project focused on adapting Washington State University's Phytophthora identification protocol into a version in which high school students can use. Using biotechnology tools like PCR and electrophoresis, we hope to create a step-by-step process that allows Bellarmine students to identify Phytophthora species locally. In the past, this project has been done before. However, sample collection has not been done here at Bellarmine. Oftentimes, sample collection has been done at Washington State University.
As for our problem, Phytophthora is a plant pathogen that's especially difficult to identify accurately because of its symptoms often look similar to those caused by other plant diseases, such as particularly Pythium, which is something we'll mention later in our project. This overlap in visual symptoms makes it easy to misidentify infections, which can lead to ineffective treatments or misunderstanding of how widespread the pathogen actually is. Additionally, most current tracking and identification efforts focus on nurseries and agricultural settings. This means that the distribution and range of Phytophthora in natural or community environments, such as forests, parks, or unmanaged land, are often harder to monitor and go unnoticed. On to our objectives. The main objective of this project was to develop and refine a Phytophthora identification protocol that is both accurate and accessible to high school students. While Phytophthora identification protocols already exist at the professional level, they are rarely designed for or implemented by high school students, making this approach relatively unexplored. Another goal was to implement molecular biology techniques, specifically PCR, gel electrophoresis, and DNA sequencing to identify Phytophthora species from environmental samples. By adapting these techniques to fit the high school lab equipment, we aim to make the more advanced pathogen identification more feasible outside of research universities. Additionally, since this is a methods piece and we're simply testing the methods to make sure they work in a high school setting, we do not have any hypothesis other than making sure our methods work in the long run. This project was divided into two main parts, DNA isolation and DNA identification. The primary focus of this project was DNA isolation, as this step had not previously been successfully performed at Bellarmine. DNA identification, while more precise, had already been partially explored, but not fully validated in this lab setting. During our sample collection, we took both soil and water samples from Snake Lake in Tacoma, Washington. Each sample was taken from the base of alder trees, as those are most often infected with Phytophthora and serve as bait for a host of infections. Several methods were used in order to bait the soil and water. We will only focus on the full leaves used, as those were the most isolated. The leaves were used as a more isolated bait for the Phytophthora to bond with, as stated in the Washington State University sampling procedure. The samples were then left to wait for 5 days and Phytophthora could attach to the leaves. After the samples were collected, they had to be plated to allow for easier extraction. The 4 steps shown were taken directly from the Washington State University sampling procedure. The steps were designed to help allow the leaves to be infected and prevent other species from infecting the samples. The steps in order were leaf bagging, disc plating, microscope analysis, and ultimately a second round of disc plating. First, we're going to go into leaf baiting. This step allowed for more infection on the leaves and gave the brown spots time to develop due to the moisture in the bag and the leaves serving as bait, just as the image on the screen shows. These spots were used as signs of where the infection was strongest and where we should punch out discs. The discs were punched out with a standard hole punch on the edge of the brown spots and were made ready for the next step. Now we move on to the DNA plating. Once discs were punched out, we had to make sure there was only Phytophthora growing on the sample. This was done with a part V8 plate, as shown in the image. This was infused with antibiotics, which prevented the growth of bacteria, but didn't hinder any Phytophthora growth. These were allowed to grow for about a week or so before moving on to the microscope analysis. Once the plates had been given time to grow, we analyzed them underneath the microscope. One of the 2 images on the screen is of Phytophthora, and the other is of Pythium. While I'm describing this step, see if you can figure out which is which. We looked for Phytophthora in the smaller, denser growths, as that is how Phytophthora spreads, rather than being much more spread out like Pythium. Once the Phytophthora was identified, with its branching structure and denser growth, we cut it out with a sterilized scalpel and placed it onto a V8 plate where it could grow more isolated. And now, as I'm sure most of you have already guessed, the image on the left shows the Phytophthora that we were able to successfully cut out of our sample.
Now for our DNA extraction piece, a small sample of agar containing the organism was taken from the V8 plate and placed into a 100 microliter tube. The samples then went through a series of steps, including incubation, vortexing, and centrifugation to break down cell walls and release DNA. After cooling to room temperature, the samples were frozen to preserve the DNA for later analysis. This step ultimately proved to be the most challenging part of the protocol. Now for PCR, or polymerase chain reaction, is a technique used to make millions of copies of a specific DNA region so it can be analyzed. We prepared a master mix containing primers and other chemicals that provide the building blocks for DNA replication. The primers we used were ITS6 and ITS4, which target the ITS gene region, a common DNA barcode used to identify Phytophthora species. PCR takes place in a thermocycler, a machine that cycles through specific temperatures to allow DNA replication. We use the Mini 1 system, which is a smaller and more affordable thermocycler than those used at Washington State University. Although it has lower output, it performs the same function and allowed us to adapt the protocol to a high school lab setting. After PCR, the amplified DNA was analyzed using gel electrophoresis, which is used to visualize DNA and confirm that the correct gene is amplified. PCR produces millions of copies for the gene, so it can be analyzed more easily, as stated previously, but gel electrophoresis then confirms whether this PCR was successful. Because DNA is negatively charged, it moves through an agar gel towards the positive end when an electric current is applied, with smaller fragments targeting farther than larger ones. Our gel contains 6 wells, a DNA ladder for size comparison, a negative control to check for contamination, and 4 sample walls. Samples that showed clear bands at the expected size were planned to be sent to an external sequencing company where the DNA sequence was compared to other known databases to see how closely it matched other Phytophthora species to confirm its identification.
And now on to our results. So we were able to narrow down what went wrong with our Oh wait, I'm sorry, I've got a little ahead of myself there, so I'm going to have to backtrack a little bit. Initially, no visible DNA bands appeared during the electrophoresis. This indicated that something in the protocol was not working correctly. At this point, we shifted focus from troubleshooting to troubleshooting rather than just testing the same steps. We were able to narrow down what went wrong with the DNA extraction PCR, and we were able to narrow down what went wrong with the DNA extraction, PCR, and electrophoresis. This is because underneath the microscope, we were able to tell that we had been able to successfully collect the sample of Phytophthora due to it being visible underneath the microscope. In order to isolate the issue, we collected samples from Washington State University that had already gone through all steps except for PCR and electrophoresis. These samples were then tested under controlled conditions. We collected 2 samples from Washington State University, one that had all steps except for electrophoresis, and one that still needed PCR and electrophoresis. The middle image shows the sample that had PCR already done, and it showed a clear band all the way to the right. This matches the 3rd level on the ladder used for scale, and it was about 500 base pairs long. This matched the expected range that we were looking for and showed that it was a successful use of electrophoresis. Next, we moved on to the one that still needed PCR and electrophoresis. The final image shows that sample, and it had multiple different bands that were clearly present. This means that it wasn't as pure of a sample through our PCR, but there is still a clear band around the 3rd ladder, which shows that it was about 500 base pairs long, which also matched our desired sample.
Overall, the protocol was partially translated successfully to a high school lab setting. PCR and electrophoresis were confirmed to work reliably here at the Bellarmine labs. However, final testing indicated that the DNA extraction phase was the source of failure. While this step remains unresolved, identifying the specific problem is a critical outcome of the project, and it provides us with a clear direction for future refinement. This work demonstrates that identifying Phytophthora through DNA-based protocol like PCR and electrophoresis is possible here at Bellarmine.
We had various limitations in our project which both impacted our scale and timeline greatly. First, due to Bellarmine not having the majority of the resources needed for our project, we had to order our chemicals online, which cost us multiple weeks of testing and limited the number of experiments we could do. Additionally, at Snake Lake, the water level lowered greatly in the summer months, which made it impossible to collect water or wet soil during the entire summer. Our scale impacted us also, as it made so we didn't have as many attempts at the procedure and couldn't as well account for smaller unavoidable errors. Finally, even though we tried 3 methods, 2 failed, with the discs not isolating the phytophthora enough, and the leaf bag we used was just stolen in the weeks that we were waiting for it to be collected. And as the image showed, that's the leaf bag we tried to put in Snake Lake, but when we went back about 2 weeks later, it was just gone for no reason.
For future research, the first pathway is to retest the DNA procedure shown in this project and figure out either what went wrong or prove that it's possible to undergo DNA extraction here at Bellarmine. Second, once the Foley is perfectly executed, students can send a DNA sample in to get sequenced and compare it to the BLAST, or Basic Local Assignment Search Tool database, which compares the sequence to local and known Phytophthora species. Additionally, students can complete the procedure on a larger scale, such as an entire younger or underclassmen marine chemistry class, in order to gain a better understanding of the Phytophthora presence in Snake Lake. Finally, future students can compile all of the steps laid out in this project into a manual in order to finalize the procedure, along with DNA barcoding for that identification process. For our acknowledgements, we have a few people we'd like to thank and shout out. First of all, Mr. DeGroat and Ms. Nuno, Dr. Marion Elliott, who has provided us with invaluable guidance throughout this project. We'd like to thank our family and our friends, and most importantly, the Marine Chemistry class of 2026.
Citations, and are there any questions for us? It's over there. I'm sorry, could you repeat that a little louder? So the question was, how is the protocol different from the Washington State University research, is that right? So our protocol is different mainly in the scale of our experiment. As we mentioned, we use the Mini-1 system for our procedure. That only has a few wells, whereas a normal electrophoresis machine would have a much larger amount, like about 50, which caused it to be a much larger experiment. Also, the steps were very simplified the way we did it, rather than focusing on more complex stuff. So we were able to collect this stuff individually, rather than just doing everything individually at the lab, and the resources that we had made it pretty different.
And to add on to what Liam just said, we also differed in terms of how we chose to get our Phytophthora samples. We took multiple different methods to try to infect our rhododendron leaves with Phytophthora. Sometimes we would bathe the leaves in water or soil. Sometimes we would let it dry, or just let the leaf discs stay in. However, ultimately, our most successful approach was when we let the rhododendron leaves stay in the water and dry it out for a period of time, allowing the Phytophthora to grow. Are there any other questions? And we'd like to introduce Mr. DeGroat. He has an announcement which I bet you all have been waiting for.
We're going to take a break. And there's some treats outside, about 10 minutes or so. Stretch your legs, stretch your mind. I'm going to invite Lauren Pickles to come on up, and she's going to speak to you guys about red tide, although you don't use that term, do you? But about saxitoxin. So come on up and tell us about your research.
Hello everyone, my name is Lauren Pickles and my project is an evaluation of saxitoxin levels in marine invertebrates from the Chukchi and Beaufort Seas. Starting off, what are saxitoxins? Saxitoxins are a kind of paralytic shellfish toxin which cause paralytic shellfish poisoning, which can affect all levels of the ecosystem. Paralytic shellfish poisoning is a relevant issue and has been linked to various die-offs of several species, including northern fur seals off the coast of St. Paul Island in 2024. Paralytic shellfish toxins, and saxitoxins specifically, are dangerous as they are sodium-blocking neurotoxins which inhibit the transfer of information throughout the brain. Lastly, saxitoxin is caused by harmful algal blooms, also referred to as HABs. More specifically, blooms of the dinoflagellate Alexandrium catanella are the most common source, especially in marine ecosystems. So why is this important? As I mentioned, saxitoxin has profound effects on the environment. Several related die-offs have been reported, and even if concentrations aren't enough to be fatal, they still significantly reduce the health of animal populations. Additionally, saxitoxins can affect people, especially Arctic indigenous communities who rely on larger animals for subsistence purposes. Worsening harmful algal blooms and saxitoxin presence is climate change driven. Harmful algal blooms are unique in that they rest in cyst beds on the sea floor and can survive for up to a century until water conditions are favorable to bloom. This means that as sea temperatures are rising, blooms are becoming more extreme and more frequent. Even though the Arctic is pretty far north from us, the fact that saxitoxins are so prevalent there is concerning and serves as an indicator for the overall health of the ocean. Here in the Pacific Northwest, we occasionally get large harmful algal bloom occurrences, and warming global temperatures are not helping this. To touch on some previous research, a study by Anderson in 2014 did a lot of background research on saxitoxin. The study provides a great overview of all information related to saxitoxin and other paralytic shellfish toxins. This paper helped me gain a really solid understanding of all of the general information and gave me a good basis. A study by Dr. Lefevre in 2022 studied a spike in saxitoxin concentration in the Alaskan region during the summer of 2019, which was anomalously warm. The study concluded that these warmer sea temperatures and jump in saxitoxin levels were correlated. This is evident in toxicity levels above the U.S. Food and Drug Administration's regulatory limit of 800 nanograms per gram. Lastly, a study by Boyvenru in 2021 established a guideline for the ideal water conditions for HABs. The study found that typically algal blooms do best at temperatures ranging from 9 to 19 degrees Celsius, and the study also looked at a couple other parameters, including salinity. The tolerance of harmful algal blooms is noted here, as they can thrive in a range of conditions. So the overall purpose of my study was to evaluate the presence of saxitoxins in clams and worms in Arctic regions and its relationship to factors such as time of collection, location, and variation in sea temperature. And I did this by analyzing concentration compared to collection month and location, and then comparing observed toxin levels with corresponding water temperature data. My first hypothesis was that August in the Chukchi Sea would have the highest concentrations because of warmer temperatures in the summer months and the presence of a large cyst bed in the Chukchi Sea. My second hypothesis was that higher water temperatures would correlate with elevated saxitoxin concentrations in both clams and worms, as found by previous research. To touch on the role of student and mentor,
I worked over 5 different sessions during the summer of 2024 at a NOAA lab at the Northwest Fisheries and Science Center in Seattle, Washington. I worked under Emily Bauer specifically, who was the lab technician, and she worked me through all of the different lab processes and helped me gain a solid understanding. And then Dr. Kathy Lefebvre was the one who runs the lab in general, and so she got me in the door. Moving on to my methods, and starting with sample collection, clams and worms were collected from cruises in July, August, and November of 2022 by NOAA teams. And these samples came from the Chukchi and Beaufort Sea regions off the coast of Alaska. The samples were kept frozen until we processed them at NOAA's lab in the summer of 2024. For toxin extraction, this process happened in 2 stages. The first is aliquoting, which involves dissecting and removing a portion of each animal to be measured. So first, we measured the clam's length and width with the shell on, and then we removed the animal portion, took its mass, recorded that, and placed each sample in its own test tube. And then worm length and mass was also taken, and again, each sample was placed in its own test tube. The second stage was the actual toxin extraction and ELISA. So first, samples were homogenized with methanol, and this is because saxitoxins themselves are hydrophobic. So when the methanol is added and the samples are blended, the toxin will leave the sample and sit in the methanol so that it can be removed later. Next, we ran an ELISA, which stands for Enzyme-Linked Immunosorbent Assay. This sounds very complicated, but it really isn't. Ultimately, what ELISA does is use antibodies to find and measure a specific substance, in this case saxitoxin. So what this kit gives us are results in the formable range of colors, which then have to be placed in a plate reader in order to get the colors into a unit of concentration that can be measured. And then finally, we took that unit of concentration and put it into a computer program called Prism to get it into the unit that we use, which is nanograms per gram. Lastly, for temperature data, I collected temperature data through a NOAA dataset to find mean monthly sea surface temperature for each of the locations in which the samples were collected. And I got the specific locations by looking at the latitude and longitude of each distributed biological observatory station where the samples were collected and plugging that into the dataset. So here's a map of the region in general, the Chukchi and Beaufort Seas off of the coast of Alaska. And then the red lines indicate the approximate locations of the stations in which the samples and temperature data came from. So to look at my results, starting with saxitoxin concentration by month, and this is also broken down into both species, clams and worms. Starting with July, I had a clam mean concentration of 118 nanograms per gram, and a worm mean concentration of 124 nanograms per gram. August had a clam mean concentration of 210, and a worm mean concentration of 18.5, and November had a clam mean concentration of 585 nanograms per gram, and a worm mean concentration of 21.5. Across August and November, the clams had significantly higher levels of concentration compared to worms. July was not like this, however, and there are a few limitations I will touch on later. Additionally, November demonstrated the highest concentrations across all 3 months. It is also important to note that all 3 months had samples above the Food and Drug Administration's regulatory limit that I mentioned earlier, which is alarming as this level is extreme, especially in a region so far north and cold. Next, to look at saxitoxin concentration by location. July had a mean concentration of 122 nanograms per gram, but only had samples from the Chukchi Sea. August had a Chukchi Sea mean concentration of 307 nanograms per gram, and a Beaufort mean concentration of 26 nanograms per gram. And lastly, November had a Chukchi mean concentration of 533 nanograms per gram, and a Beaufort Sea mean concentration of 93.2 nanograms per gram. As with concentration compared to month, August and November had much higher levels than July, and November had the highest overall. Between the 2 locations, the Chukchi Sea did demonstrate higher levels than the Beaufort Sea. Lastly, looking at saxitoxin concentration by temperature, The data was pretty scattered here in terms of these two factors, and no statistical significance is shown. So there are a range of temperatures and pretty high concentrations no matter where it falls on that scale, which was unexpected. So as a quick reminder, the main objective of this study was to evaluate the presence of saxitoxins in clams and worms in Arctic regions and the relationship between various factors such as temperature, time of collection, and location. My first hypothesis that August in the Chukchi Sea would have the highest concentrations was partially supported by data. Saxitoxin concentration compared to month and location both had statistically significant results with a p-value less than 0.01 throughout August and November, which differs from the hypothesis that August alone would have the highest concentrations. Additionally, November showed even higher concentrations than August, which was unexpected. However, the Chukchi Sea did demonstrate significantly higher concentrations than the Beaufort Sea. The likely reasoning for this is warmer temperatures in the summer months and the flow of currents, as well as the presence of a large seabed in the Chukchi Sea. My second hypothesis that higher water temperatures would correlate with elevated saxitoxin concentrations in clams and worms was not supported by the data. Saxitoxin concentration, when compared to temperature, did not demonstrate significant results. There appeared to be no correlation between these two factors, and the data was pretty scattered. This differs from previous research, which has demonstrated a relationship. However, that did not appear in this study specifically. Next, I want to go into a few of the limitations in this study. The biggest was sample size. July, August, and November had sample sizes of 23, 37, and 35 respectively. This limits the legitimacy of data and makes it hard to generalize results to the larger ecosystem in the region. Additionally, July had the smallest sample size and only contained samples from the Chukchi Sea, which further hindered the ability to make comparisons across all 3 months. The likely explanation for this is timing of the summer cruises, which occurred in late July and carried into August, meaning they didn't have time to collect samples they got out of the Chukchi region before they were able to collect samples from the Beaufort Sea as well from July. The final limitation relates to sea temperature data and that it should have been collected from the benthic zone on the ocean floor, so the deepest part of each region. This type of data was not accessible, however, as it is extremely difficult to collect from the necessary depth. Therefore, the temperature data used was sea surface temperature, which may impact results as the concentration levels came from organisms at deeper depths. Future research could explore this relationship more within species higher in the food chain. Additionally, there is room to look deeper into concentrations in other locations farther south, as there are known seabeds in other regions. Ultimately, it is important to continue to track toxin presence as it is expected to worsen and poses a threat to the health of the world's marine ecosystems. For my acknowledgements, I'd like to thank Emily Bowers for working with me in the lab, Dr. Kathy Lefevre for getting me in the door, my parents for being super supportive throughout this whole process, and lastly, the Marine Chemistry class of 2026 for making it a memorable 4 years. Are there any questions? The question was, do I think having a limited sample size in July and if that impacted the results? The answer is I definitely think it did. I think that the general trend that the July data had lower levels than the August and November data would have continued even if the sample size was bigger, but it did make it difficult to make accurate comparisons across all 3 months like I was intending to. The question was, why was the Chukchi Sea chosen? The easiest explanation for this is that it is one of the locations where they were collecting the data. So they're not collecting samples from all over the ocean. There are specific locations.
And the other reason is that it does give a good comparison, especially with the Beaufort Sea, since it's farther south, so temperatures are slightly warmer. And it's also much shallower than the Beaufort Sea, which makes it easier to draw comparisons. Can you repeat the question, please? So I think the question was that just confirming that I used the sea surface temperature data instead of trying to get it from the deeper depths. Is that right? So also if I could have gotten something a little bit more accurate to the temperature at deeper depths. And the answer to that is not really. I looked everywhere and data isn't really out there that was accessible to me as far as temperature from that deep of depth. They don't collect it very often, so it wasn't something that I could use. And so I had to use sea surface temperature data, which as I said can impact the results as it would be warmer than where the organisms came from. But there isn't really any way that I know of to model specifically the lower temperature in relation to the concentrations that I had. Next, I want to introduce Anya Moravec to talk about AI.
Thank you all for taking the time to be here. I would like to start with a little bit of an interactive activity. So could you please raise your hand if you've ever used AI? Now keep them raised if you would trust AI playing a role in the communication that you have with your physician. If you notice, a majority of the hands around you have dropped. Hi, my name is Anya Moravec, and my project is on evaluating the preferability of artificial intelligence in the electronic health records messaging system. The electronic health records system, also known as EHR, was originally created in the 1960s as a way to move paper files online. In the early 2000s, they were modified to allow for secure messaging between patients and their healthcare providers through different portals. Since 2018, there has been a threefold increase in patient portal messaging. This is closely linked with the COVID-19 pandemic, as it became one of the limited ways that patients could interact with their doctors. And the rate of messages never returned down to pre-pandemic rate levels. This is according to a study by Michael A. Hansen and others in 2024. Recently, physicians report spending an average of 52 minutes a day responding to their EHR inbox. However, this does vary between specialties. This is closely linked to physician burnout, as they have to spend unpaid hours responding to their inbox. This increased inbox burden is also associated with the reduction of physician-patient interaction, which impacts the quality of care that patients receive. Many EHR companies have started to implement an AI chatbot to solve some of these problems. For my study, I looked at three main review of literatures. They included Cavaliero and others in a 2025 study, which used a survey to find that participants preferred messages that were written by physicians compared to AI once they figured out which messages were written by physicians or AI. Another study I looked at was Lizor Ritz and others in a 2025 study that used a survey for an AI scribe program, and it showed general openness to the program. However, there was more skepticism seen with younger patients. Another study I looked at was gun lack and others in a 2025 study, which used semi-structured focus groups and regarded the potential of AI in the medical fields, and it was gained positive support, but it was only positive towards the usage of AI as a support tool and not as an independent decisions-making system. This brings me to my first problem, that there is a gap in knowledge regarding how patients are acceptable to the usage of AI that is replacing direct and indirect messages with physicians. My first objective is to analyze comfort levels via a participant survey that compares comfortability levels of messaging between AI and physicians. And my first hypothesis is that participants will be highly accepting of AI-assisted communication with low-stakes tasks. My second problem is that it is unknown whether a patient's familiarity with technology will affect how they prefer to communicate with their health providers. My second objective is to examine the correlation between participants' comfortability with technology and their preference on the role of AI in their communication. My second hypothesis is that participants with higher self-reported comfort with technology will demonstrate significantly greater preference for AI involvement with their healthcare communication. I mentored under Dr. Philip Louie, an orthopedic spine surgeon, a medical student Kenneth Gwen at Virginia Mason Franciscan in Seattle. Kenneth and I developed the first draft of our survey, which Dr. Louie then revised and distributed, and all of us participated in data analysis and interpretation. However, we did our analysis independently of each other. The survey was distributed to 1,075 participants, and the questions were made on a 6-point Likert scale as seen above, where 0 shows not at all comfortable, while 5 is very comfortable. It was composed of around 60 questions, taking less than 10 minutes. There was 5 different sections, and I looked at the first 3 sections, the first of which was general information, the second was comfort with technology, and the third was attitudes and preferences towards AI in healthcare. The survey was administered through the company RedCap, and participants gained monetary rewards from completion. So the average age of participants was around 45 years old, with plus or minus 16 years in the range. A little bit over half of the participants were female, and a little bit under half of the participants were male. Around 78% of the participants were white, and the second majority of participants were Black or African American, being 14%. Around 88% of the participants have received an education higher than the high school level. So this graph displays the average answer to the question, how much do you prefer each of the following communication approaches? The one in red displays whether patients are comfortable receiving information first from an AI chatbot, while the blue bar displays direct interaction from a human provider from the start. The AI first had a significantly lower average, around 2 points lower, and a smaller 10% error bar range when compared to the human first range. The figure on the left displays the distribution of each question, with its answers on the Likert scale, where the lighter the color is, the closer the answer is to 0, while the darker it is, the closer the answer is to 5. The figure on the right demonstrates the average of each question, with error bars for the 10% range of the average, and each of the questions line up, so question 8 on the far left is also question 8 on the graph on the right, the farthest left. The colors in red are the questions regarding AI preference, while the questions in blue ask about human preference. All the questions regarding AI have low ranges, around 2.2 to 2.5, which points towards low preference towards AI being used in healthcare. All of the distributions of these answers are also distributed on parabolic shapes, which shows that the answers lean towards participants not preferring the usage of AI. Participants show that they do not prefer receiving health information and or updates from an AI chatbot, which is seen through question 8. Participants showed distrust towards health information that is provided by an AI system, which is seen through question 9. Overall, most participants said that they do not think that AI tools are that effective in helping patients understand their diagnosis or condition, which is seen through question 10. And question 11 asked if participants would prefer using an AI chatbot if it provided faster results, which most participants said that they do not prefer that. Question 12 asked about participants' comfortability with participating in a two-way chat with their healthcare provider, for questions or concerns regarding their health, and question 13 asks how important it is for the participants to have a real person available for conversations regarding their health. Both question 12 and 13 show an exponential curve with the majority of participants choosing 5. They also have averages above 4 and also have larger 10% error range. The data also has a p-value of less than 0.01, which means that the data is statistically significant. The graph on the left shows the distribution of each answer. The darker the scale, the closer the answer is to 5. While the graph on the right shows the same answers, but it shows the averages for each question. Question 17 asks if participants think that healthcare quality could overall improve through the implementation of AI, which was met with non-preference and had an average a little over 2.6. The distribution of this answer is demonstrated by a parabolic curve, which shows that most participants do not think highly of the way AI can improve healthcare quality. Question 18 was met with overall high comfort levels, and it asked if participants would want a human provider to explain how and why AI is used in their care. Question 19 asked if participants were concerned about privacy and security when AI is involved in communication with their care, which most participants said that that is a concern when they have AI implementation. The majority of participants said that AI should be a tool for support and not should replace overall human insight. And question 21 said that asked participants if they should have options to choose between AI and human-like communication, which was met with the highest average. These questions help show how the majority of participants do not prefer the usage of AI and how they are concerned with how and when AI is being implemented in their care. The data also has a p-value of less than 0.01, which means that the data is statistically significant. This shows the average communication role preferences, with physicians being seen the most and it decreasing linearly. There is a major gap between care coordinator and AI chatbot, which shows that participants do not prefer the usage of AI chatbot communication. This graph helps show the average answer to how helpful it would be to interact with an AI chatbot for each of the following. These are non-clinical questions that should not require physician oversight. They include questions about scheduling appointments, which had the highest averages, to discussing minor symptoms or concerns. This helps answer my first hypothesis because I predicted that participants would be comfortable with AI when it's used for smaller tasks, which is not seen by this graph. This graph displays the average answer to the question about how helpful would it be to speak directly to a human provider for each of the following situations. The main reason why I wanted to include this graph was to show it as a comparison of answers from this graph to the previous graph that asked about AI usage. This graph shows the correlation between average response and comfortability with technology, which is displayed by the x-axis, and the average response of comfort with the usage of AI, which is displayed by the y-axis. So the bigger the bubbles displayed, the more responses were made at that point. For example, if someone rated themselves an average of 3 with technology and comfort with AI, they would be, that's where they would be on the graph. And the more responses that were made with that point, the bigger the bubble would be. Most of the bubbles are located around the 4 to 5 range of comfortability with technology, and there's a large range for comfortability with AI. The data has a p-value of less than 0.004, which means that the data is statistically significant. The data also has an R-value of 0.16, which means that there is a positive correlation, however it is a weak correlation. So the data shows that there's a big difference between preferences towards human contact and that of AI. AI had low averages even for non-clinical tasks or smaller tasks, which disproves my first hypothesis. There was a weak correlation, however still a positive correlation, between technological comfort and preferability of artificial intelligence, which supports my second hypothesis. So my problem was that with the healthcare world changing and AI becoming more developed, it is unknown how acceptable patients are to AI being used when receiving their care. So I did a large diverse survey to get the opinions of participants' preferability on the usage of artificial intelligence. I found that there was very high preference towards human contact and lots of discomfort towards AI being used. What this means is that AI is not preferred by the majority and transparency of AI usage in healthcare is needed. Some of the limitations of my study include that participants might not have answered some of the questions truthfully, and others that participants may not understand the aspects of the survey. For example, they might have been confused on the Likert scale. Participants could have over or underestimated their ability to use technology, and also prior experience with AI is also unknown. Some future research following this project is a survey that is sent to physicians that asks them how much they trust AI chatbots being used in communication with their patients, and their opinion of the role of AI in the EHR inbox managing system. Another future research is a survey sent to patients who have used an AI chatbot compared to patients who have solely communicated through human providers to see if their opinions differ after experience of how useful AI can be in the medical field, specifically managing EHR inbox. I would like to thank my mentors, Dr. Philip Louie and Kenneth Glenn, for giving me the opportunity to work with them on this project. I'd like to thank my family for always being there for me. I'd like to thank Mr. DeGroat and Ms. Nuna for helping me throughout my time in Chem and for planning and making the Hawaii trip an amazing experience. I'd like to thank Mr. Rayberger for letting me borrow his computer. And lastly, I would like to thank the Marine Chem class of 2026. I had the best time spending the past 4 years ago. Questions? Yeah. So the question was if I looked at education as an idea of the correlation between AI, is that correct? Yeah. I didn't look at that because part of the reason was I didn't get the specific person and their high school level. They just gave me the average, the complete amount. So they just said that 88% seemed higher than an education higher than the high school level. So I didn't have the information to be able to do that. So the question was, how much were people paid? Very little. I don't think they made a lot of money from the survey. How do I feel about AI? I think that it's a very good support tool. I think that it should be used as long as it's used for transparency and then it's overseen by human providers. I would now like to invite Christopher and Langdon to talk about eelgrass.
Our project is mapping the area and density of Sostera Marina at Titlow Beach Marine Preserve. Our project is mapping the area and density of Sostera Marina at Titlow Beach Marine Preserve. My name is Landon McClurg.
And I'm Christopher Lowe.
Eelgrass has been declining across the Puget Sound for many years. According to Puget Sound Info, total eelgrass coverage decreased by about 6,000 acres between 2014 and 2023. Globally, researchers have linked eelgrass loss to several factors. One major cause is climate change. Fraser et al. Found that rising temperatures can reduce photosynthetic efficiency, limit growth, and increase vulnerability to disease. Another cause is eelgrass wasting disease. According to Brackle et al., disease causes black lesions on eelgrass leaves and reduces their ability to photosynthesize. Competition from invasive species is also a factor. Lord et al. Explained that eelgrass can struggle to reproduce when other species outcompete it. Together, these factors all contribute to the ongoing decline of eelgrass.
Now, why is eelgrass so important? Eelgrass is a marine angiosperm species that is also a keystone species, in which ecosystems largely depend on its role in maintaining the stability for nearby wildlife. Eelgrass beds are a vital source of food and shelter for marine life. Many young fish use these beds as a nursery due to the rich and dense areas for food and protection. Without them, fish lose critical nursery habitats to grow and reproduce. The beds also help stabilize the seafloor, in which roots stabilize the sediments through a system called rhizome mats, which help prevent erosion by anchoring roots to the ground. Again, without eelgrass, coastal erosion would increase due to the lack of rhizome mats. Eelgrass also store greenhouse gases like carbon dioxide. The main purpose of this is to help maintain oxygen levels inside the water to help protect marine life. If eelgrass populations decline, a lot more carbon would exist in the environment and impact temperature, water quality, and much more. Lastly, eelgrass is frequently known as an indicator species due to its sensitivity to changes in the environment. For example, the absorption of excess nutrients. For example, excess fertilizer from an incoming runoff would rapidly increase the eelgrass density and could indicate that excess nutrients are entering the water.
In 2012, marine chemistry students conducted a study at TIPO to investigate the relationship between eelgrass density and distance from the shoreline. They found that eelgrass was denser the closer it was to shore. They also created a GPS map showing what the eelgrass bed looked like at the time. However, this study was done more than a decade ago, so it is unclear whether the eelgrass bed has changed since then, or if the pattern of higher density near the shore is still true today. Another factor that may affect these findings is the nearby culvert that flows into the eelgrass beds. According to the Salmon Recovery Portal, the culvert carries lagoon water into the eelgrass beds. Because of this, it is uncertain whether eelgrass density is influenced by distance from shore, the culvert flow, or both. By examining the current density and distribution of eelgrass, we can establish a baseline and better understand whether there is a relationship between eelgrass density and the culvert.
We identified 3 main gaps. First, it is unknown if the size of the eelgrass bed had changed, as it has been over a decade since the last created map. Second, it is unknown if there is a relationship between eelgrass density and its proximity to the culvert. Lastly, it is unknown if the previously found relationship of eelgrass being denser when closer to the shore is still true to this day.
So we had 3 objectives. Our first objective was to create a new map of the eelgrass bed at Titlow Beach. Our second objective was to record the density of the bed based on distance from the culvert and from the shore. And our final objective was to compare our data with previous research to notice any trends or changes.
We made 3 hypotheses. First, we hypothesized that the size of the eelgrass bed would decrease, following the decline in total eelgrass coverage in the Puget Sound. Second, we hypothesized that eelgrass density would be greater the closer it is to the culvert, as we believe that something could be inside the water that is affecting the eelgrass population. And third, we hypothesized that density would be greater the closer it is to the shore, continuing the correlation found by previous marine chemistry students in 2012.
So for our methods on creating the map, we used the Gaia GPS app on a smartphone to map the bed. We walked along the edge of the eelgrass bed, visually identifying where the eelgrass ended and the bare ground began. As we walked, we recorded GPS points every 5 to 10 feet and took photos with location data attached. The negative 4.3 foot low tide gave us access to the intertidal zone, and we used a paddleboard to map the deeper edges of the bed.
Our mapping method created this visualizer, representing the main eelgrass bed as a continuous polygon. This bed is concentrated in the central region of Titlow Beach, and we use this to create 7 different zones. Zones 1 through 4, circled here, measure distance from the culvert outflow parallel to the shoreline, in which Zone 1 is directly on the edge of the outflow, and Zone 4 is far away from the culvert. Zones 5 through 7, circled here, measure distance from the shore in a perpendicular direction from the shoreline, in which Zone 5 is the closest to the shore, while Zone 7 is the furthest point. We want to keep note that for the purpose of this study, we excluded the smaller isolated patches surrounding the main bed in order to make data collection easier. The real eelgrass beds include many patchy areas that are not shown on this visualizer in all 4 directions.
We collected density data in May, when eelgrass typically reaches its peak biomass. To measure density, we followed the Jennifer Rusink lab protocol and used a 25 by 25 centimeter PVC quadra. At each zone, we randomly placed the quadra on the ground and counted all the eelgrass shoots inside it. We repeated this process 3 times at each zone to get an average.
Moving on to our results, we found that eelgrass density decreased as we moved farther from the culvert. Zone 1, which was the closest to the culvert, had the highest density at 24.4 shoots on average. Zones 3 and 4, which were the furthest zones away from the culvert, had the lowest density at 14.7 and 17.7 shoots on average, respectively. Zone 2 had an average count of 23 shoots and visualized a trend of density decreasing when moving away from the culvert.
Regarding distance from the shore, density was greatest in the zones closer to the shoreline. Zones 5 and 6 had the greatest density at around 24.7 shoots, while Zone 7, which was farther from the shore, had a shoot count of around 16.4. This visualizes a trend of density increasing when the zone is located closer to the shore.
Now for our results, or for our discussion, revisiting the map we created for this study, we found that our first hypothesis is inconclusive. We were unable to find an accurate way to compare the new and the older map. One reason for this includes the older map lacking precise coordinate points that stop us from being able to overlay our new map with the older one. Additionally, our visualizer does not accurately represent the entire eelgrass bed. We only recorded the bed that was continuous and connected to the culvert outflow, leaving out numerous amounts of patchy areas nearby that surrounded the bed in all 4 directions. There are also other areas in the Titlow Beach that have eelgrass populations that are not measured, leading to an inconclusive answer to our hypothesis. Although we have no definite answer, future work could be done to answer this hypothesis, such as improving the method used to collect data points, and also including the patches that were not accounted for in our data.
Our results supported our second hypothesis that eelgrass density would be higher when closer to the culvert. Zone 1, located directly inside the culvert, had an average of 27.4 shoots, while zones 3 and 4, the farthest from the culvert, had an average of 14.7 and 17.7 shoots. This means that eelgrass was more dense when in closer proximity to the culvert. Although our data suggests a potential correlation, it was not found to be statistically significant. One possible explanation for this is that there may be runoff from the culvert that could be adding nutrients to the water that promote eelgrass growth. Future research could include testing the water chemistry from the culvert to see if there are unusual nutrient levels. Another direction for future research includes repeating the study with an increased sample size to improve the reliability of the results.
Finally, our third hypothesis was supported, in which eelgrass density was higher when in closer proximity to the shore. Zone 5, the zone closest to the shore, had the highest average of 24.7 shoots, while Zone 7, the farthest from the shore, had an average of 16.4 shoots. These results follow the same trend identified in the 2012 study, in which eelgrass density increased when approaching the shore. Although our data suggests a potential correlation, it was not found to be statistically significant. A possible explanation for this is that eelgrass at shallower depths receive more sunlight than those at a deeper depth. This means that eelgrass located closer to the shore would have less depth and more sunlight to support their growth, in comparison to eelgrass that is deeper and further away.
Our study had several major limitations. First, our map did not include smaller patches that were separate from the main eelgrass bed. This made it harder to determine the full extent of eelgrass at Titlow. Future studies should focus on mapping these patchy areas. Another limitation was that we did not collect water chemistry data, so we could not determine whether the culvert was affecting eelgrass growth. Finally, we collected all of our data on a single day. Because of this, we were not able to examine seasonal or long-term trends. Future research could collect data over a longer period of time to better understand changes in eelgrass growth.
And then for our acknowledgements, we would like to give a special thanks to Mr. DeGroat, our family, and the Marine Chemistry class of 2026 for making this all possible. Here are our citations. Any questions? Are there any questions that need to be answered? Yes.
You said the old data did not have coordinate
So the question was, did the old map not have any coordinate points, but did we include coordinate points in ours, correct? The answer for that is yes. Every 5 to 10 steps, we collected a GPS map point, and we collected all of those to create that polygon in the visualizer earlier. Yes?
How deep is the water in the deeper?
So the question was, how deep was the water in the deeper edge of the bed? So at the time we collected the data, it was a negative 4-foot tide, so it was about 6 foot deep at the back edge of the bed.
Yes. I'm sorry, could you repeat the question? So the question was, can this information be used elsewhere or only at Titlow? Unfortunately, our results only apply to Titlow Beach for now. Future studies could possibly explore this correlation to see if it is statistically significant, to see if this trend could apply to other areas. Yes?
Is there multiple bluegrass beds at Titlow or just that?
So the question was, are there multiple eelgrass beds at Titlow Beach? So we mapped the main continuous bed, but the bed kind of continues off farther. It's just pretty patchy, so we didn't include that in our map.
How do you expect the eelgrass population fluctuating in like other parts of the world, like based off decrease right now?
I'm sorry, do you mind repeating that?
How do you think that the eelgrass population will continue to grow like in other areas of Washington?
So the question was, how do we think the eelgrass population will fluctuate in other areas? Is that correct? Yes. The answer for that is we do not know. Our project only applied to Titlow Beach exclusively, but further research could be done for different locations to see if they have similar correlations to what we found.
Next up we have Calvin Rubio, and he's going to talk to us about forever chemicals.
Here, we see a glass of water. You may think you know what's in it, but chances are there are things in your tap water that you don't want in it. Your tap water most likely isn't just H2O molecules. It could have all sorts of chemicals, microplastics, and bacteria making it their home. Among these contaminants are a group of harmful chemicals known as PFAS, or forever chemicals. They're found in a broad range of materials in household appliances. Some examples are pictured here. Hi, I'm Calvin, and I'm going to be talking to you about granulated activated carbon versus zero-valent iron in PFAS forever chemical remediation. But what is PFAS? PFAS stands for per and polyfluoroalkyl substances. They're found primarily in the defense and mining sectors, in plastics and firefighting fluids. They can be very harmful, causing cancer and other serious health issues down the line. When first discovered, PFAS was viewed like some sort of miracle substance, a lot like asbestos. It has high heat resistance and exhibits hydrophilic and hydrophobic properties. These miracle qualities, unfortunately, make it very difficult to remediate in high quantities. The structure is characterized by a central carbon backbone saturated with fluorine atoms containing at least one functional group. There are a few major subgroups, but the ones I want to focus on specifically are perfluorooctanoic acid, PFOA, and perfluorooctane sulfonic acid, PFOS. These are the two most prevalent subgroups in drinking water. Why does PFAS matter to us? Like I said earlier, PFAS can be found in common household items, such as non-stick cookware, paint, or waterproof clothing. Because of this prevalence, it's critical that remediation techniques are established to remove any potential drinking water contamination. For a review of research, in a January 2025 paper, Figueres et al. Outlined the link between PFAS and cancer, as well as other adverse health effects. This was one factor leading to the establishment of guidelines and regulations by the EPA and other regulatory organizations. In addition, a 2021 paper highlights the difficulty with PFAS remediation and how it stems from the simultaneous hydrophobic and hydrophilic properties and the high heat resistance. And finally, a 2025 news article from the Stevens Institute of Technology stipulates that iron powder could outperform carbon in particulate filtration. On the topic of filtration, let's talk about two major filtration materials. The first one is granulated activated carbon, or GAC. This is used in commercial filters, such as Brita, and filters out impurities, such as chlorine, which is responsible for the taste and odor of tap water. It works well because of a large surface area, so there's a lot of space for the water to interact with the carbon. The Elite filter is also certified, the Brita Elite filter, is also certified to remove contaminants and microplastics. Microscale zero-valent iron, on the other hand, MZVI, refers to iron in its elemental state and is used industrially to destroy contaminants through redox reactions. The iron reacts directly with contaminant molecules and donates electrons, changing the contaminant to a less harmful substance. In some cases, the contaminant is then removed through direct absorption with the iron, though this happens more with heavy metals than fluorocarbons. The current gap in knowledge is that not enough research has been done about the comparison between GAC and MZVI in the context of PFAS remediation. So my study aims to compare the effectiveness of GAC and MZVI in remediating specifically PFOA and PFOS. I hypothesize that the MZVI will outperform the GAC and remove more of the PFAS from the sample. My role was to design the experimental setup, conduct background research, and analyze the resulting data. My mentor, Mindy Graden, critiqued the experimental setup, reviewed the writing section, and oversaw the experimental procedure. The PFAS sample was acquired from Wellington Laboratories in Ontario. Pictured is the original concentration of both PFOA and PFOS. Each PFAS subset was shipped with a concentration of 250 nanograms per milliliter. The 1.2 milliliter sample was diluted to 1,000 milliliters, so the concentration translates to about 300 nanograms per milliliter. The EPA has set an enforceable limit of 0.004 nanograms per milliliter. My study aims to discover which treatment method can lower the concentration to as near 0.004 nanograms per milliliter as possible. The procedure consisted of two trials. One with the Brita Elite filter, which primarily uses GAC and is rated for PFAS removal, and one with the microscale zero-valent iron in a homemade filter. The MZVI filter was constructed using polyethylene terephthalate glycol, or PETG, as it contains the least amount of PFAS of any commercial plastic tube. Two coffee filters were attached to the end of the tube with rubber bands to ensure the iron powder remained in the filter system during filtration. The mass of filter material in the Brita filter was measured to determine how much MZVI was required. That mass was then placed atop the coffee filters to create the filter system. For the actual procedure, the water was passed through the filters and collected in separate pitchers before being transferred to the containers used for shipping and analysis. The pitcher on the left contains the MZVI filter, with its sample containers pictured in front. The pitcher on the right has the Brita Elite filter, and its respective sample containers pictured in front of it. After the testing phase was completed, the samples were sent to Eurofins Scientific for HPLC analysis. HPLC stands for High Performance Liquid Chromatography, and it involves injecting the solid sample into a mobile liquid phase, then passing it through a stationary HPLC column to separate the components into distinct bands, which are then read by a computer to give the concentration of each individual component. Now for my results. Eurofins Scientific reported back on over 40 PFAS subsets. Some of the most notable are pictured here. The initial concentration for all subsets was 300 nanograms per milliliter. As shown in the two rightmost columns, remediation was largely successful for the carbon and less successful for the iron, although the PFTRDA, PFTEDA, and PFDOS all saw high amounts of remediation using both methods. Here's a more succinct data sheet with just the PFOA and the PFOS, along with the perfluorododecyl sulfonic acid, which is PFDOS, which I found interesting because of the dramatic decrease in concentration in both trials. Here on the left, we see the concentrations of each of the three aforementioned PFAS subsets at the start and after testing with the granulated activated carbon. As can be seen, the concentrations decreased by at least 150 nanograms per milliliter for each of the three PFAS subsets. On the right, we see the concentrations of each PFAS subgroup before and after treatment with the zero-valent iron. Treatment proved largely less significant than the carbon, although still remarkably high rates were seen with the PFDOS. My hypothesis, again, was that the zero-valent iron would outperform the granulated activated carbon in amount remediated. This was not supported by the data, as the iron appeared to have a far less significant effect on most PFAS subsets. Interestingly, however, both the carbon and the iron had a strong remediating effect on the perfluorododecyl sulfonic acid, which was unexpected as it does not line up with the other PFAS subsets. My main limitation is that my filter setup for the zero-valent iron was insufficient for this particular filtration experiment. Generally, zero-valent iron is injected directly into contaminated soil to react in situ. But my project just had the contaminated water pass through the iron. In addition, the iron actually, sorry, that's incorrect. And then, since the mass was used as a control, the quantity of iron was actually far less than the quantity of carbon. This means that the water came in contact with far more carbon than iron in the actual remediation process. For future research, I would compare the Brita Elite filter, which is what we used, to the Brita Standard filter, which is not rated for the removal of PFAS, to determine how much of an improvement the Elite filter is over the standard. In addition, microscale zero-valent iron was used in this experiment. There are many other sizes of CVI, which serve a range of purposes unique to their particle size. A future test could compare different sizes of CVI specifically in the remediation of PFAS. And finally, I'd like to look more into the PFDOS and figure out why it saw such high remediation rates in both trials, even when the iron appeared mostly ineffective against most of the other subsets. For some thank yous, I'd like to first say thank you to Mr. DeGroat for helping me through my whole project. I'd also like to say thank you to my mentor, Mindy Graden, and to my family for keeping me going, and of course to the Marine Chemistry Class of 2026. You guys are so great. It's been a blast. Are there any questions? Okay, the question was, is there PFAS specifically in Tacoma and Gig Harbor water? Is that correct? So we actually are positioned fairly close to a like oil drilling site, which I believe has actually reported some PFAS runoff. So as far as I know, it's being dealt with, but it does matter to us because there is some potential of contamination from some of the nearby areas. Yeah. Okay, so the question was, what is the difference between PFDOS and PFOS? So they're just different types of PFAS, basically. PFDOS stands for perfluorododecyl sulfonic acid, and it has to do with the structure of the molecule. And PFOS is perfluorooctane sulfonic acid. Anyone else?
In your diagram of where PFAS comes from, I saw it had popcorn bags. Is that a specific part of it?
Yeah, so the question was, in that graph we saw PFAS comes in like popcorn bags. Yeah, that is a concern. Actually, it comes in a lot of fast food packaging. So when I went and did this experiment, I had to make sure not to come into contact with any like fast food packaging beforehand. But yeah, that's something that we need to avoid as well. Anyone else?
More. Right here.
I understand that at least like the road filter is like plastic. Do you think there's any like affordable solution for people who want to like filter their water but also like microplastics that you can yourself?
Okay, so the question was, is there any danger of contamination from the actual plastic Brita filter itself? Yeah, so Brita goes through a lot of lengths to try and remove as much cross-contamination as possible. So what you will see is likely low levels of contamination, but sometimes it is unfortunately unavoidable when dealing with many types of plastics. So short answer, hopefully low contamination, but there's definitely still a possibility. And now I'd like to welcome up Bridget Nuno with a very special announcement.
Thank you, Calvin. And I'm going to invite all of us to come up. I'm Bridget Nuno. Sorry, one more little presentation. I'm Bridget Nuno. I work with the fabulous Mr. Dave DeGroat, and I have been doing this with him for the past 9 years, but he's been doing this for the past 23 years. 26 years? Sorry, I wrote about that one. Okay. But we are just so appreciative to him. And as a way to say thank you, the Marine Chem class of 2026 has created something I know you wanted, another slideshow.
Alright, so how we're going to do this, we're going to just give a little thank you to you for everything you do. I'm going to start off with a thank you for your humor. It's definitely helped the past 4 years. We've had some tough times, we've had some tribulations and trials, and making sure that you were always there, cracking a joke, making fun of me for being cold, making fun of all of us for doing something wrong. It's definitely helped us through the projects and through the past 4 years, so I really appreciate that.
Yeah, again, I want to second that, especially during senior year. All the SNLs we watched before, they were amazing, although some of them were not very appropriate. They were awesome. And I want to thank you for perfecting us, going through about 15 drafts of the presentation before giving it, and you've just been amazing. Just an absolute confidence boost and just an amazing addition to the work.
Okay, I'm going to speed this up. Thank you for letting us bake in the sun for hours upon hours in Hawaii. I had a great time.
Yeah, thank you for really helping this process go smoothly, helping us every step of the way. It's really appreciated. Thank you.
Thank you for your cheers every class, which makes it super exciting.
Thank you for teaching us the importance of walking backwards and how it's helpful for our brain health by making us walk around campus backwards.
I would like to thank you for putting up with our late wake-up call the day that we flew out of Hawaii. I'd like to thank you for all the support and guidance you've given all of us throughout all our projects and our papers.
I'd like to thank you for answering our many questions on the Hawaii trip about your professional DJ career.
I want to thank you for always showing up for us, especially when we need you most, and especially for your donuts junior year when we had to get to school really early for our meetings.
I want to thank you for helping me with the corals. You sent me like 5 articles. That was really helpful. And trusting me with the keys to the computer lab for the image day that I probably shouldn't have taken the computer for. But thank you.
Thank you so much for your care and your empathy. I remember in Hawaii, you gave me medication so I didn't have to throw up on the boat, and I really appreciate that. Thank you so much.
I'd just like to thank you for all your help with the research process, and I'd also like to thank you for warning us about the dangers of bladder infections.
I want to thank you for always being able to lighten the mood with your sarcasm, and I hope you enjoy the retirement home.
I would like to thank you for always pressuring us to keep doing our work even when we didn't really want to, and making sure we always knew what we had to do and when we had to do it.
I would like to thank you for helping me to discover I was allergic to seasickness medication. And I hope your future scuba diving adventures are much more smooth sailing than most of ours were.
Thank you, Mr. DeGroat, for showing me how to have good posture. You can see how horrible my posture looks in that image. I am very inspired by how you stand up straight all the time, as you can see on the right. And in the future, make sure you go to Denny's for that senior discount.
Not even Mr. DeGroat could teach me to have good posture. However, he could teach me to make sure to wear more sunscreen. Some of you might remember from Hawaii, I changed color to purple. That's not my natural color. Never again. Thank you.
I would like to thank you for saving that family of deer from the wildfire, and for saving us when the Shark Tank, the bars dropped and you had to save us from the sharks, and also for climbing Mount Everest, truly a great feat, and also for being the best driver on the Marine Chem trip.
I'm going to change your grades tomorrow. Well, thank you very much. I do have some thank yous as well. Thank you to this class, my last class here, as a matter of fact, because I'm retiring after this year. And the people taking over are Ms. Nuno, Ms. Eder, Mr. Top, and so they'll be left in good hands. But I'm so thankful that you guys were my last class. I'm probably still going to be battling all these things when they need it, but I'll be coming back. A lot of other people responsible for setting things up in here, our communications team, our ed tech folks, administration, supporting our program overall, and then certainly parents. We appreciate the support that you guys and the love and the care that you provide for your students, and entrusting us with their care. And for the most part, I got to say, at least 65% of the time, I cared about them. So that's pretty high. In any case, I believe we are finished. Typically, we take some pictures, and maybe this is the one you want to take a picture in front of, perhaps, for anybody who's interested. But the rest of you, sophomores and freshmen, don't forget about your assignments. Juniors, don't forget about your assignment as well. And make some appointments with me before I leave. and thank